Hello, AP Biology students. We're going to go over the sixth section in Unit 4. And in this section, we're going to talk about something called cell cycle regulations. In this specific section, we're going to talk about how the cell cycle is going to be regulated so the cells don't continuously go through mitosis. Certain signaling molecules are going to be received by the cell to either initiate the cell cycle or to stop the cell cycle. This is specifically why the AP or College Board organizes cell communication within this unit, because this is a part of the cell communication content where cells are signaling to each other in order to either initiate mitosis to start or to stop. I again have moved the cell communication content to Unit 2 because I like it to fit in with that unit and the just cells overall. We refer to these signaling molecules as growth factors because they're initiating mitosis to occur. Some of these molecules that are going to initiate different parts of the cell cycle are called cyclins. These are just internal signaling proteins that either increase or decrease during different times of cell division. There are four main types of cyclins, and in AP biology, you don't really need to know any of the specific types of these types of cyclins, but you should have a general idea of what cyclins do. You can see the different types of cyclins as they increase and decrease and what they initiate in this graph. So basically, these are just molecules that are going to start different processes within the different steps of the cell cycle. But it's important to remember that they don't act alone. They're going to act with something called CDK. CDKs are cyclin-dependent kinases. Remember that kinases are enzymes that phosphorylate other proteins. And usually that phosphorylation is going to activate them. And like the name implies, these are going to be kinases that are dependent on cyclins. These CDK enzymes are always going to be present in the cytoplasm. However, they're not going to be activated until certain cyclins are going to be attached or bind with them. This is going to initiate different aspects of the cell cycle because these cyclin molecules or cyclin proteins are going to initiate the kinases to activate other proteins. So basically what I'm trying to get across here is that these CDKs are going to bind with cyclins during different time periods of the cell cycle to initiate the certain processes that need to go on. Here's an example of what I'm talking about. You can see the CDK above is inactivated on the left-hand side here because there's no cyclin around. Whenever that CDK binds with a cyclin, it's going to initiate or it's going to phosphorylate other target proteins. These target proteins, as you can see, are going to complete some process within the cell cycle that needs to occur. But these CDKs will only activate these proteins when the cyclins are present because, again, the CDKs need to have those cyclins around. Here's an example of the M cyclins, just to give you an idea of what they do. Again, they're going to bind with CDKs that are specific for this step. These CDKs are going to initiate the process of proteins making spindle fibers. They're also going to initiate proteins making the nuclear membrane breakdown. And they're also going to initiate proteins making the chromosomes condense. These are all things that occur during M phase. So the CDKs and the M cyclins are going to bind together to initiate proteins to make all of these things happen. You can see in the example here, the CDK binding with the M cyclin to initiate the breakdown of the nuclear membrane. And we got to end on this one. It's just perfect. CDKs only are initiated when they bind with their cyclin molecule. Absolutely great.